Mom, can I start?
Okay, let me. Mohan.
Yes, we can start now.
Shall we start? Okay. Adira, start.
Good morning, everyone, and welcome to the fourth day of International Webinar Series, BioSpectrum 2021, on the recent trends in biological sciences, organized by the Postgraduate and Research Department of Microbiology, Sri Shankara College, Kaladi. Today, we are having the Technical Section 4, led by Dr. Biju B. Thomas, Assistant Professor of Ophthalmology Research, USC Roski Eye Institute, University of Southern California, Los Angeles, California, and the topic of today's session is stem cell-based techniques to restore vision. Dr. Biju has completed his master's degree in zoology from Mahatma Gandhi College, Trivandrum, Kerala, and he acquired his PhD in behavioral neuroscience from University of Kerala, Trivandrum, India. Earlier, he worked as lecturer at St. Stephen's College, Pattanapuram, and also at St. Birchman's College, Changanasheri, Kerala. Currently, he is working as an assistant professor of ophthalmology research, University of Southern California, Los Angeles. Before we begin, I request all the participants to mute your microphones and turn off your video for the smooth conduct of the session. I would like to invite Dr. Sumi Mary George, Associate Professor, Postgraduate and Research Department of Microbiology, to deliver the welcome speech. Ma'am, please.
Good morning. A warm welcome to the Technical Section 4 of BioSpectrum 2021. Today, we are lucky to have Dr. Biju B. Thomas, an eminent scientist in the field of ophthalmology research, as our speaker on the topic stem cell-based techniques to restore vision. Currently, he is working as assistant professor in ophthalmology research, University of Southern California, Roski Eye Institute, Los Angeles, USA. His area of interest are new treatment strategies for retinal degeneration diseases, retinal transplantation for cell replacement therapies, retinal prosthesis for vision restoration, retinal degeneration disease models for testing therapeutics, new methods and approaches for reliable measurement of visual functions in rodents. So as Adira added, he is a Master of Science in Zoology from Mahatma Gandhi College, University of Kerala. He has PhD in Behavioral Neurosciences, Department of Zoology, University of Kerala, and teaching and research experience. And to add more, he has also many awards and honors to his credit. He is a recipient of Young Investigator Travel Grant Award 2002, Bright Focus Foundation Award 2016, and he is a principal investigator and co-investigator of 12 funded projects. Noteworthy among them are the 4 ongoing stem cell projects funded by National Eye Institute, NIH, and California Institute of Regenerative Medicine. He is an holder of 3 patents, and his long list of publications proves his subject expertise. He has published 42 referred journal articles, 2 in 2021, 1 is in press, and even COVID-19 pandemic couldn't stop him, where here all have almost stopped. Also, he has publication in referred reviews. Then he had chapters, editorials, clinical communication like case reports and letters, abstracts and presentation, etc. So we are very much excited to hear you, sir. Once again, I welcome you, sir. Thank you.
Thank you.
Thank you.
Great.
Thank you, ma'am. Now, Dr. Biju B. Thomas, the dice is all yours.
Okay, so thank you very much for the great introduction. And also, I would like to thank the organizers, especially my long-term friend, Dr. Mohan, so for giving me this opportunity to talk to you. And also, I personally believe that when especially my background from Kerala and going forward with my research interest, accomplishing a lot of goals and achievements. And also I was fortunate to work with the world famous researchers and inventors, and including some of these products that are already in clinical trials. So I started from basic science to a very productive translational research. So that is a very lucky opportunity for me to see that from bench to bedside. So that is what we say. So because when I started my research, I was mostly, my thinking, imagination about research was mostly based on basic research where we are looking at very fundamental things of science. But when after I reaching US, I came to understand that the research what we find in the beginning is not something useful unless we are able to translate it into a clinical application. Anyway, more than that, I would like to give you my experience, which is mostly on stem cell research. So I began my research in behavioral neuroscience from the University of Kerala, where I was mostly working with different rodents and also trying to understand the influence of orthopedic changes on the behavior of the rodents. So that was a very helpful background for me, because that's from where I moved to eye research, because of course the light from the environment goes through the eye and control various physiological activities of the body. It's not just for vision, but there are a lot of other things that physiology of the animal is involved with the environment. So when it comes to eye, that is what I am now more focused on, especially the diseases that are affecting the eye. I might need to give you some fundamental idea about the structure of the eye before I move to my presentation, because there are a lot of things that you may not understand if I suddenly jump into the topic. So I will give you some introduction about the structure of the eye and what are the area of my interest. Can you see the screen now?
Yes, sir.
Okay, so yeah. So this is the slide which I am showing is a structure of the eye, and sorry. Okay. So when you look at the eye, consider it as a like a spherical globe or maybe a football with a hole on the center. That is the front side. That is our pupil, the hole through which the light goes into the eye through this pupil, and then so the light fall into the back of the eye. So there is only a small area through which the light can enter into the eye, and it goes into the back of the eye. So on the right side, there is a fine structure of the enlarged structure of the eye, the retina, which is the sensitive area of the eye. So the light falls into the photoreceptors, which is the rods and cones, which is further inside the retina. So when you see the, I don't know if you can see my cursor or not. Can you see it or not? Can you see the cursor?
No, sir.
Okay.
We can.
You can see the laser point now.
It's coming.
You can see it, right?
Yes.
now we can see.
Okay. So this is a small portion from this area, a small portion of the retina that is enlarged here. And here you can see that the photoreceptors are this area. This is a photoreceptor layer, and the various neurons in the retina are more into this area. And on the back of the photoreceptors, you can see a thin layer, and that layer is called the retinal pigment epithelium. So there are three different major structures of the retina which contribute to visual perception. Of course, the photoreceptors comes first, the major. If there are no photoreceptors, you cannot see anything. Then comes the neurons here that transmit all the information from the photoreceptors into the brain, and the retinal pigment epithelium, that is single layer, that is responsible for supporting, nourishing the photoreceptors, because one of the highly metabolic areas of the body is retina. So it's always highly active. So it needs a lot of energy, and there is always chance for more physiological adverse effect happening to that area because it is highly active. So when the light from here goes through the eye, passes through the different layers, passes through all these layers and reaches the photoreceptors. And the photoreceptors are then converts that light energy into electrical signals that will pass through these neurons. And all the neurons will terminate into a bundle of nerves that goes into the through the optic nerve and goes to the brain. So that is how the eye functions. Here you can see two different major photoreceptors, rods and cones, and I'm not going into the details of these photoreceptors. Just above the photoreceptors, you can see the 4 epithelial cells. Those are the, it's not 4, but here it is shown as 4. Retinal pigment epithelium, which is highly supporting cells. And outside just above the retinal pigment epithelium, you can see another pink layer, and that layer is the blood vessels. So basically, nutrients goes through the blood vessels into the retinal pigment epithelium, and the retinal pigment epithelium produces certain biological factors and also transmits nutrients from blood into the photoreceptors, and also all the waste materials formed here, because these photoreceptors, they have an area which always shed, just like our skin is peeled off. Tip of the photoreceptors are always continuously replaced. So there is a lot of accumulation of potential for a lot of accumulation of waste there. So that waste need to be taken by the RPE and then pushed back into the blood vessels so that it can be removed. So what I'm telling in detail here is anything that happens to any of these components, what I discussed here, like the neuronal retina, or the photoreceptors, or the retinal pigment epithelium, or even the blood vessels, that can adversely affect the eyesight. So that's what we call like a eye disease. So generally people say that somebody is losing vision or some acute or chronic eye disease, more like a vision loss. That can be due to any of these layers. And one really disappointing thing about these kind of diseases is it's very difficult to cure them. There is no treatment. There is no medicine that you can either intake or drop, like a drop, or even injecting into the eye is not going to cure it, because you are damaging, the cells are getting damaged, or the photoreceptors are dysfunctional, or if they are not producing the right kind of light sensitive biological factors that need to be converted into electrical signals, or if the neurons are not functional, or if they are degenerating, that is another problem. Then the disease can also affect the optic nerve that goes to the brain. If anything happened to the optic nerve, there are several diseases, like, I don't know, you might have heard about glaucoma, where there is a high pressure inside the, because this is a fluid filled space. If the pressure goes up, it will constrict the optic nerve, and that will also cause diseases. So there are a lot of different chance for diseases that can affect the eye. Then that's a general disease about the eye, but there are genetic disorders called retinitis pigmentosa, which is a kind of disease that affect the phototranslation cascade, the chain of reactions that takes place inside the photoreceptors, leading to conversion of light signals into electrical signals. So in that case, there is a failure of photoreceptors. There is another disease called age-related macular degeneration, where I'll show you in the next slide, but that's another condition where the retinal pigment epithelium is damaged. So it cannot support the photoreceptors. So in that case, what will happen is photoreceptors may not get sufficient nourishments. Retinal pigment epithelium is not clearing out all the debris there, so that will develop a toxic environment there. So in whatever situation, if there is a change in the normal homeostasis of the retina, the immediately affected section of the retina is the photoreceptors. Photoreceptors are highly susceptible to degeneration. So in any condition, the first thing will happen is the rod photoreceptors will degenerate. I don't know if everyone know about it. There are two types of photoreceptors, rods and cones. In rodents or in nocturnal animals, it is a rod-dominated retina because the rods are highly sensitive to light. So you can, these animals, because they need to move around in the night, they need to make use of little bit of light they get. So that's why they have a rod-dominated retina. Whereas in humans and other animals that are active in the daylight, they have a more cone-dominated retina, where the cone photoreceptors are sensitive to bright light. But on the other hand, there is some disadvantage for rod response, because even though it is highly sensitive, what will happen is you cannot see color. The rod is only one type of photoreceptors, whereas cones there are blue cones, green sensitive cones. There are different types of cones that will contribute to color vision in the normal light, whereas in the dark, probably you might have experienced it when you are in a dark room, lights are turned off, you can little bit see things around, but you are not able to see any colors because that is contributed by the rods. So when the degeneration happens, some diseases, the rods will degenerate first. In some other cases, cones will degenerate first. In some other conditions, both rods and cones will degenerate. But the problem is, even if the rods will degenerate first, it will affect the cones eventually. So it will also cause lead to blindness. Next slide, I'm going to show you so this is a normal condition. You know, the image on the top is the round spherical moon-like structure it's called a fundus picture. So when after you dilate your pupil and use a special camera to take a picture, this is a human fundus. What you see inside is what you see here is so macular region is the highly so the macular region is the area which is having lot of sorry, I have to, looks like something happened. 1 second, I have to upload it. Can you see the slides or no?
You need to put it again.
No, sir.
Okay, so I think I have to load
Share
it.
again.
Yeah. So this first one is a normal eye where you see, it's just showing that how kids are holding two balls and how you can see it in a person with a normal vision. And the second one is a condition called other diseases called retinitis pigmentosa, where you will mostly lose the peripheral vision, whereas your central vision it's called a tunnel vision. You can see, the person with this disease can see the face of the two boys, but they cannot see the periphery. This is because of the type of disease generally called as retinitis pigmentosa. And in the next case, that's a totally, here you see just opposite to what you see in the first case. So you can see that everywhere it is highly, very dim vision, but you can see some peripheral areas are better, whereas the central vision, you cannot see the face of the kids, but you can see only the hands, and they're holding the balls. So these are the two different types of disease which we are dealing with. One is retinitis pigmentosa and other one is age-related macular degeneration. The difference between these two is in retinitis pigmentosa, it is mostly due to mutation of the rods or cone photoreceptors. So when a mutation happens, it will fail to produce the required proteins that is required for in a normal vision. And in that case, because of the abnormal development of the photoreceptors, they will degenerate and lead to a vision loss. So, but in the age-related macular degeneration, as the name says, age-related macular degeneration, the central area which is more important for acute vision is less functional because of the retinal pigment epithelium failure, as due to the old age. Especially this is more common among people who smoke or eat unhealthy food or lack of exercise. All those conditions that can lead to earlier cell death in the retinal pigment epithelium. So retinal pigment epithelium failed to support the photoreceptors. And because it is mostly related to age, is commonly found in people above the age of 60, and the macular area is more damaged. So you are losing the central vision, but you can see a little bit in the periphery. So we call it as age-related macular degeneration. So these are the two common diseases found in humans. But there are a lot of different subtypes of, especially for retinitis pigmentosa, there may be like 20 different types of retinitis pigmentosa. Depends on what type of cells or what type of protein is dysfunctional. So in either case, the only solution in these conditions are either to replace the lost cells or bypass the lost cells. And also, I just wanted to mention you other conditions where you can damage retina is using laser beams. That is another reason for damages happening to the eye. So the possible approaches for vision restoration once the photoreceptors are damaged is on replace or restore the lost retinal photoreceptors, or bypass the damage. So there is a difference between bypass and replace. Replace means a direct replacement of the damaged cells. So the only one way you can replace the damaged cells is to introduce new healthy cells. So that is what the importance of stem cell therapy is. In the last decade, there is a lot of ways you can generate cells. Before it was more derived from embryonic stem cells were converted into retinal cells, differentiated and into pure retinal cells and used for transplantation. More recently, there is induced pluripotent stem cells where a patient's own cells can be collected, stem cells can be generated from like hair follicles or some other sources. So these cells can be cultured and converted into retinal cells. So by using these cells, the approach is to deliver these cells into the diseased retina, hoping that you will be able to replace some of the dead cells. So basically, you're introducing new healthy cells that will go there, attached to the host retina, survive, mature, integrate with the host retina and sometimes even make connections with the existing neurons. So that is called the replacement method. Then bypassing the damaged retina is by using prosthesis devices. So I worked on with a group where we developed the first retinal prosthesis that is used for patients now. It has a lot of limitations, so I'm not going into the details of retinal prosthesis now because it is a totally different and highly elaborate topic that need to be discussed and need more time. So anyway, I'm keeping it aside, the retinal prosthesis, which is of course a great scope for blind people, where basically you put a electronic, or I would say that a multi-electrode array into the neurons of the retina, and there is a camera sitting outside. That camera will connected with your glasses, and it will find out the images and transmit it into the electrical signals that are converted directly stimulate the inside neurons, and that will pass into the brain and you get a visual percept. So the problem with that is, there is the resolution is low, the pixel size is low because of the limitations of making the electrodes. You cannot put 10,000 or 100,000 electrodes into the eye because there is a difficulty to manufacture those kind of tiny electrodes that can fit inside the retina. But on the other hand, for getting the right kind of resolution or pixel size, you might need maybe like at least 1,000 electrodes. But the maximum we could make was like 60 electrodes. That's only our capability, or the current capability with the science to produce the electrodes. I mean, of course you can make, but you have to make it really tiny to fit inside the eye. So that's one limitation of the retinal prosthesis. So we are looking at other options, and that's why I'm more interested in replacing or restoring vision by introducing healthy cells. And there is the third and final type of approach for curing blindness is make remaining non-light responsive retinal neurons light sensitive. So that's a nanotechnology approach where you can introduce certain nanoparticles that are sensitive to light. So these nanoparticles are delivered into the eye. They will go and attach in all different types of cells inside the retina. And once, it's just like it works like photoreceptors, but it has also a lot of limitations. So several laboratories have tried it, showed some initial positive results, but still not in a clinical trial yet. So the only approaches that are currently in clinical trial are cell replacement and retinal prosthesis. Of course, the retinal prosthesis moved from clinical trial to commercial level. So one company is actually producing the prosthesis, but its popularity is not very high because of the already mentioned limitations. So if anybody have any questions, I mean, I don't mind answering it right now, but maybe it's up to you. So now coming to the transplantation, that is again, when you deliver cells into the eye, for example, in a layman's language, I would just say that stem cells are injected into the eye, for example, simply saying stem cells are injected into the eye. How these stem cells work? So many people hear about stem cell therapies, and I don't think many people know about the real mechanism by which the stem cells work. There are different mechanisms. The application of the stem cells is very wide, and I strongly believe that it's going to play a very critical role in the future in the health sciences. We are still seeing only tip of the iceberg when we talk about the application of stem cells. There is a long way to go. So how the stem cells work, it can work in different ways. When you deliver cells into the eye, for example, a diseased eye, where there is already damage to the photoreceptors, probably damage to the neurons, damage to the retinal pigment epithelium, so completely messed up, or the eye is not functioning. In that case, when you inject or deliver stem cells into the eye, it can work in two different ways. One is trophic effect, and second one is called a direct replacement effect. So the difference between trophic effect and direct cell replacement effect is when the healthy cells, especially the stem cells, has the potency to release a lot of neurotrophic factors. I don't know if you ever heard about brain-derived neurotrophic factors, BDNF. It's a very common and probably a terminology that everyone might have heard about. Brain-derived neurotrophic factor is a factor that is released by many of the cells, and it is highly supportive to unhealthy neurons. So stem cells has the potential to produce some of these or many of the neurotrophic factors. So the way the neurotrophic factors influence the disease condition is still not completely known, but it is kind of a rejuvenation process. When these neurotrophic factors are released into a diseased neurons or photoreceptors, they can give some a short-term energy or rejuvenation. So they will, at least they will temporarily stop the degeneration process, and it might also restore some of its lost function. That will give an immediate effect. A patient who get a stem cell injection probably will say that I got some improvement in the vision, but it may not last for long because the trophic effect is a transient effect. Stem cell cannot secrete all these neurotrophic factors forever. So it might happen for a couple of months or maybe a year, then it will start to diminish. Then the second, so that is a very common thing that many cases when we say that stem cell therapy is showing some positive effect, it could be a trophic effect. So the good thing is there is no requirement for integration or connections, but it is transient, short-term benefit. But in a direct replacement effect, what will happen is that is what we are looking at. For example, we differentiate photoreceptors, make retinal type cells, and transplant it into the diseased retina or underneath the diseased retina. Okay, 1 second, please. Okay, and then hoping that the transplanted cells will integrate into the host retina. So there is a difference between trophic effect and replacement effect. When we say replacement effect, the cells what we transplant should perfectly stay there, survive there, and it need to connect with the host retina. Otherwise, it will remain as a foreign material. So that is very challenging, but we already found there are strong indication that there are formations are there. So these kind of connections are the determining factor for a long-term benefit of stem cell treatment. So I'm just showing some of so how we find out all this after transplantation. Basically, this is from a animal model where human fetal retina was transplanted. So I worked with a group long back where the ethical rules and regulations were not very strict. So that time, early 2000, there was opportunity to get aborted fetus, collect the retina, and then there were some experiments in which one of the group I collaborated, they transplanted that into the rat eye and to show how the transplant looks like. And so both the human fetal retina and also animal fetal retina both were tried for transplantation. So this is kind of a proof of concept that shows that you can transplant into the diseased eye, and the transplanted retina can remain there as a healthy sheet. 1 second. Okay, so I'm trying to find out my cursor anyway. I don't see it. That's okay. So this is one of the recent approaches for retina transplantation. So I don't know if you heard about retinal organoids. So more recently, techniques, stem cell based techniques are developed for making retinal pigment epithelium, differentiating retinal pigment epithelium from stem cells. And more recently, there is a way you can culture stem cells in a culture dish, and provide the factors that will help the cells to develop into a retina. So it is called eye on a dish, or culturing. You're making it appears very impressive. So basically, you are making an eye in a culture dish using stem cells. So the advantage of using that is called retinal organoids. The advantage of using the organoids are because there are no ethical issues. You don't have to depend on a fetal retina for transplantation. So you can make several pieces of these kind of sheets for transplantation in a culture dish by proper differentiation of the stem cells and making it into spherical structures comparable to an eye. But don't think that it is size of the eye. Nobody can make an eye in a culture dish, because these kind of organoids are tiny, maybe the size of a mustard seed. That's the maximum size it will grow. But if you cut it and look at it in different layers, you can see that it has pretty much everything you find in a adult retina. Of course, it will grow. When you culture here, you can see that different time points, D23, D31, day 42, and day 48. So it's like the embryogenesis happens in all animals, but it's not like a complete organ culture. But it shows some of the similarities with the mature retina in terms of different types of cells present there. So this is a new technique where you can develop a retina on a culture dish, and that can be used for various experiments. So when you make it in small dishes, you can dissect it using a micro scissor, and you can use these pieces for transplantation into the eye. When I talk about transplantation, just always think that it is animals, rats or mice, not in humans. So this picture shows, this is called OCT, or optical coherence tomography. It's a way of scanning the eye. You can use a camera-based system where you can see the different layers of the, so it will vertical scan. So in the, if you recollect the first figure I showed, it showed the different layers of the eye with the retina, photoreceptors, neurons, and back of the retina, RPE, and also the choroid part. So that was a diagrammatic sketch, but using a non-invasive live imaging technique, you can pretty much see everything, what we saw there. This is a retina, and if you look at the two sides, you can see that it is extremely thin because it is degenerated, and the middle area is more like a bulged area. That is the normal thickness of a retina. So here what we did was we transplanted the fetal retina, the organoid sheets into the diseased retina to restore the shape of the retina. So this is one of the initial tests to study how the transplants work in a animal model. So these are all retinal degenerate. So when I say retinal degenerate animal models means there are rats that mimics human diseases, eye diseases. So these animals are either naturally occurred mutation related diseases or transgenic animal models. So in the previous figure, you see the imaging technique that shows there are different, the transplant is there. We also do the immunostaining. So different types, immunohistochemistry to identify different types of cells in the retina, in the transplant area and also in the host retina to find out what are the cells that are surviving, whether there is any integration with the host, between the host and transplant retina. That is what most important thing is. So if you look there on the bottom right one, it is written host and transplant. You are not seeing any kind of barriers there. You can see that there is overlap of different types of cells, including neurons, going back and forth between the transplant and the host. That is a strong indication that there is there could be potential functional integrations between the transplant and the host. So this is more detailed immunostaining images and also histology, morphological assessments showing how the transplants survive and how these transplant integrate with the host retina. What are the different, because I mean I don't want to go into the details of all the specific staining used because it's beyond the scope of many of the audience who may not be able to understand it, but just the immunostaining is for different types of neurons and different types of photoreceptors, host cells and transplant cells. So basically what we are trying to show here is there is a strong indication of integration between the host and transplant. So that is a very positive sign that there is potential for a direct replacement. So one of the most successful transplant approaches we have done is the retinal pigment epithelium transplantation. So this is one reason why in the beginning I described about the importance of retinal pigment epithelium. So the retinal pigment epithelium is on the back of the photoreceptors, and again I am just repeating it. So it supports the photoreceptor function by removing the debris and waste materials from the highly metabolic active retina, and also give nutrients into the retina for their normal functionality of the photoreceptors. So it's kind of a gateway between the outer blood circulation and the highly metabolic photoreceptors and neurons inside the retina. So the RP has a very crucial role. If the RP get damaged, like the disease called age-related macular degeneration, that can eventually lead to vision loss. So in our group, what we have done is we are one of the pioneers of differentiating retinal pigment epithelium from stem cells. And also what we did was, because the retinal pigment epithelium always remain as a monolayer, an intact monolayer. So if you inject the cells, it may not develop into a clear monolayer, because for its normal functionality and survival, it need to be transplanted as a sheet. So to make a sheet of single layer of cells, you need a substrate. So what we used was a substance called parylene. It is a non-degradable compound. It is what we used was in the next slide I will show you. So this is it's called parylene has a thickness of maybe like a couple of microns, and also in the same parylene there are thinner areas that will have even pores sufficient to allow the nutrients and materials to pass across. So when we constructed it, we made the ultra thin area with a sufficient amount. When it is really thin, there is more pores are there. So that will allow the materials to move across the membrane. So the RP cells are, so this is a parallel membrane, and this is a parallel membrane, and you can see the dots there. So on the left side, you can see on the top side is a parallel membrane without any cells, and the bottom side, the dark one, is the parallel membrane with the retinal pigment epithelium monolayer. So it is a very, even with RP, the thickness of the parallel membrane would be maybe less than 25 microns. And in animals, we have transplanted around 1 millimeter, 0.5 millimeter rectangular pieces that contains the retinal pigment epithelium as a sheet. And we transplanted it into animal models, and we found that it works fine. So in this slide, you can see more details about, if you carefully read it, you can see that polarized retinal pigment epithelium cells and ultra-thin diffusible parallel membrane. The cells are grown over the surface of this parallel membrane. And you might also notice that substrate resembling Brooks membrane. That's an additional live membrane. In the live eye, there is an additional membrane that gives support to the retinal pigment epithelium. So it comes as a single unit. So with this setup, you can have a direct replacement of the cell. So we succeeded in doing it in animal models, and then we moved to the human clinical trial. So how we find out the functionality of this retinal pigment epithelium is, as I mentioned before, the retinal pigment epithelium remove the debris from the photoreceptors. So if you stain the retinal pigment epithelium for photoreceptor sensitive antibodies, you can see that there is presence of photoreceptor debris inside the retinal pigment epithelium. So that is a direct indication that the retinal pigment epithelium is functional. So in US, we have to first establish everything in animal models, show that it survives, it functions, and also it is not causing any kind of adverse effects. So only in that case, we will be able to move to the next level, that is to approach the FDA with all this data, and we will say that, okay, this works in the animal model, so we need to do it for clinical trial. The first step would be to go for a phase 1 clinical trial where really, advanced disease condition patients will be ready. Those who are willing for clinical trial, they will come forward. So it's an expensive procedure. So once you pass the phase 1, so here we pass the phase 1, so we move to the clinical trial, and then also, so this is what the human implant looks like. This is what we used for, so it's called a CPCB RPE implant, California Project to Cure Blindness. So that particular, so here you can see that it's not a rectangle, but it's slightly, one side is oval shaped, and you can see it looks a very weird shape on one side. That is because that's where the clinician hold the implant using a forceps, surgical forceps, and then before implantation, it need to be held in his hand, and then he will put it inside the eye. So I mean, I'm not going into the details of the surgery. So basically, in the eye, what they will do is they will inject a fluid that will detach the different layers of the retina, and once it is detached, it will look like a bulged area, and there they use a micro scissor and make a cut and opening, and then with this forceps and with this membrane with a parallel, they will just push it inside and then close the eye. So it's like a 30-minute procedure for transplanting this. So this was done in advanced disease patients, and this is I'm also a co-author in this paper, but the Dr. Amir Kashani, who is the one who did all the clinical trials. So the initial trials look very good. The RP after implantation, it survived, and it is giving some improvement in the vision in some of the patients. So this is human. Again, fundus is you take a picture of the inside the eye using a special camera, and that's how you see like a moon. So you can see that the dark area inside, that is where you can even see the shape of the implant after placement in the eye. So because the retina is semi-transparent, that's how you are able to see all this. So it doesn't mean that it is just floating in the eye. It is within the different layers of the retina. That's how it is implanted. But because it is transparent and the parallel has some special light sensitive properties, that's why you see the reflection. This is to confirm and make sure that it is still surviving. So again, more pictures. You can again see the implant again in the other pictures also, how it looks like after placement in the eye. On the right side, you can see the optical coherence tomography vertical scanning pictures, again showing the position of the implant. The subject means the patient. So this is the part of the clinical trial, the initial data that was published in Science Translational Medicine. So so the previous one in patients, what we already did is transplantation of retinal pigment epithelium. Again, coming back to the disease called age-related macular degeneration, I mentioned that the disease occurs because of the dysfunction of retinal pigment epithelium. So one approach is to transplant the retinal pigment epithelium. So when you replace the healthy retinal pigment epithelium, they will give support for the photoreceptors. But we have to think about a scenario where photoreceptors are also damaged or completely lost. So in that case, there is no question of retinal pigment epithelium as such can restore vision or help to improve vision, because if in the absence of photoreceptors, there is no other supportive cells can take over its role. The photoreceptors are a minimum requirement. There should be, you don't have to have the 100% of the photoreceptors. Even if you have only 30 or 40% of the photoreceptors remaining, you would be still able to get some improvement in the vision. But when the disease is highly advanced, probably 90% of the photoreceptors are degenerated. In that case, just replacing the retinal pigment epithelium is not sufficient. There should be an approach where the photoreceptors, on the other hand, think about a situation where both RPE and photoreceptors are degenerated. So you replace the photoreceptors, but there is no RPE. Even that case won't work. It won't help because the RPE need to be there for the photoreceptor to continuously function. On the other hand, if you just replace the RPE but no photoreceptors, again, it's a failure because there is no photoreceptors to convert the light signals to electrical signals. So in an advanced disease condition, you need to have a co-graft. That is what mostly I am doing, and most of my work in my laboratory is a co-graft approach, where so just like what I described now, that is what it is. RPE transplantation alone is not sufficient in the absence of healthy photoreceptors. And the retinal organoids that we use as a source for photoreceptors, but they don't. That's one limitation of the retinal organoid. Retinal organoid is, in the beginning I mentioned about culturing the eye in a dish. When you try to make a miniature or small retina eye kind of thing in a culture dish from stem cells, most of the cells are there, which are found in a normal retina, but the retinal pigment epithelium is not growing there. The reason is because the differentiation procedure for retinal pigment epithelium is different from rest of the retina. So it's hard to get it together. So in that case, even if you consider the organoid sheets as a good source of photoreceptors, you need to get a different layer of retinal pigment epithelium and make it into a single entity, including the RO sheet, retinal organoid sheets, and the RP for successful transplantation, especially if the disease is highly advanced. So just for a diagrammatic representation, I am just showing how this. On the right side, you can see you remember that rectangular piece of RP implant, then and the green piece is a RO sheet, and so you need to have both together for transplantation when if you're looking for reconstructing a severely damaged retina. So reconstruction means when the retina is severely damaged, it will lose its old normal even shape. I'm not talking about the eye. Remember, the eye, nobody will have any loss of eye shape, even the retina is damaged. But when you look at that microstructure of the retina, you will see that it is completely messed up when the disease is highly advanced. In that case, you need to have a co-graft approach. So these are the major goals of this co-graft my research project, like how you can make the RP implants and how RO sheets and how these can be made together into a composite graft that can be used for repairing a completely diseased retina. So here, again, there is another diagrammatic representation of a diseased retina prior to transplantation. So here on the left side, A is a host retina that shows that it has retinal ganglion cells, different types of cells, and also, but it don't have any photoreceptors. But on the B, you can see a co-graft that has retinal pigment epithelium cells and photoreceptors, and also all different types of retinal cells. So if you have this, the co-graft that is A plus B, that is the C is showing, where you can completely replace all the degenerated cells. And this is an ideal situation where you are reconstructing a completely damaged retina. You can, I would say that this is more like a tissue engineering approach. So basically you are reconstructing damaged retina by using stem cell derived cells. So just showing some of the instruments which are used for transplantation. So on the left side, before I mentioned about making a bulged area where you can see that in the eye how it is made and how the instrument is used to put these transplants into the back of the eye. So this is a patented instrument, not my patency, but one of my previous mentors. So this is what is used for transplantation surgeries. So here we are just showing, previously I showed the diagrammatic sketch of the co-graft. This is a real co-graft that we made and used for transplantation. And again, the optical coherence tomography showing the thicker area, which is the transplanted area, and see the difference in the thickness, because the transplanted area of the retina, that is pretty much the normal thickness of the retina, whereas on outside the transplant area, you can see that it is really thin because it is degenerated and many of the cells are gone, so it shrink. The thickness is very small. Just showing some images of the co-graft, how different types of cells are. So here you can clearly see the area where implanted area and non-implanted area. So the co-graft implantation area, the first figure, that looks like a normal retina, where the no implantation, the control area, you can see that the retina is extremely thin. Especially the thin area is more towards the photoreceptors, where the photoreceptors are the, some of the neural retina is pretty much similar in both cases, but the photoreceptors are completely gone here. So, I mean, mostly immunostaining, and this is a optokinetic testing, is a behavior testing for testing the vision in animals. So Just giving a conclusion about the co-graft approach. So it is possible to develop retinal pigmented epithelium and retinal organoid and make it as a co-graft for retinal transplantation. So when we make the co-graft, we have to use some bioglue. So we have used alginate as a bioglue, and also we are using some fibrin glue recently. So we are trying different glues to make sure that depends on how they survive and they stay together. So based on these different techniques, we are able to make a co-graft, and we found that we can easily transplant it into animal models using the implantation tool showed before. So this approach, based on our study, this approach appears to be a very promising technique to repair a permanently damaged retina. So I would also, at this point, I would also like to mention about my collaborators, especially Dr. Humayun. He's a world-renowned retinal surgeon, and he's the inventor of retinal prosthesis. And I collaborate with the different universities. Dr. Madeline J. Saylor, she was my long-term collaborator, and once she was my mentor. And also I have support from many people, and some of the recent postdocs, I haven't mentioned their names because they were not involved in the studies. The research was supported from funding from different sources, like in the introduction it was mentioned, California Institute of Regenerative Medicine is a funding agency in California who provides support for stem cell based research, and also foundation grants, and currently I am getting support from National Eye Institute for more related work. So that's all about this presentation, and there actually I wanted to, I forgot to include that slides. Some of the things I do for vision testing is, you might, at least the people might be thinking how you know that the vision is restored after transplantation. So how we evaluate, assess the transplant, I very briefly explained about optical coherence tomography. That is a live imaging, non-invasive technique. So you can do the imaging, for example, rats, give it an anesthesia, dilate the pupil, and you can scan the retina and see how the transplant looks like. So that's one way of immediate testing of the transplant. Then after you euthanize the animal, you can take the tissue, do the histology work, do the immunostaining, and look for different types of cells. So those are the two major things that I mentioned here. But in addition to that, we also do visual behavior testing. That's one of my background in behavioral neuroscience helped me to develop different types of instruments for vision testing in rodents. That include one is a very commonly used and internationally used apparatus is called optokinetic testing, where you will put the rat in a container, and rat will be exposed to moving stripes. So if the rat can see it, it will turn its head. So we can change the width of the stripes, and when it reaches a certain level, really small stripe, the rat failed to see it and it won't turn the head. That's how another way of assessing the vision in rats. The good thing with that is you don't have to train the rat. It's an involuntary activity. Even humans, if you are sitting in a room where the walls, think about the walls has white, black and white stripes that are moving, then we will turn our head. It's called optokinetic nystagmus. Because in humans, it's easy to ask the patient, can you see it or not? But in animals, it is not possible. That's why we use this kind of visual behavior testing. Then another technique I use is, that's mostly I developed it after I came to US. So there is a visual center in the brain. So you might have heard about visual cortex, but below the visual cortex, there is a brainstem area that reaches all the retinal input. You can open the rat brain and put a multi-electrode array directly over to the top of this area and stimulate the eye with the light flashes, and you can see the responses from different electrodes, recording electrodes, and based on that you can confirm the presence or absence of retinal activity. And another very commonly used technique is called Electro Retinogram, where you place an electrode on the surface of the cornea and again stimulate the eye with light, and there are electrical activity can be recorded by that electrode. This is even used for humans in the clinic. So there are different types of techniques that we use for measuring vision after transplantation, because that is also very critical. As I mentioned before, people might think, how you know that the rat is restoring vision? So first of all, we use blind rats, so retinal degenerate animal models where the vision is completely lost. Then we transplant it with stem cells or stem cell based different products, and then we assess it with optical coherence tomography imaging, then test the vision using optokinetic testing, then use the electrophysiology, and finally euthanize the animal, take the eye, and evaluate the tissue. That's the battery of tests, and that's how we perform all the evaluations to confirm that the transplanted cells are surviving there and also it is functional. So there are also other advanced techniques what we use. We have even tried injecting a transsynaptic virus into the brain, where we found the response, and if it's called transsynaptic virus. So if the virus reaches the eye, it is very clear that there is a connection, neural connection, because the virus passes only to the synapses. So there are different tracing techniques also involved to look into these kind of things. More recently, we are looking into computational modeling, where these kind of immunostaining images, high resolution images, 3D images can be converted into computational model to see how the functionality is related with the integration with the host retina. I just wanted to mention all these things. If you have questions about anything more, I think that's it. And if you have questions, you can ask me. And anybody want to ask questions in Malayalam, also welcome.
Thank you, sir, for your wonderful presentation. Now it's time for the queries. I request the participants to post their questions in the chat box or unmute your mic and ask directly.
Hello, am I audible?
Yes.
Okay, Dr. Biju, it's one excellent presentation, very good research work. Thank you for accepting our invitation to present in the BioSpectrum 2021. And as he mentioned, he's my batchmate or hostel mate, and for about 30 years ago, and we have he's one of my very close friends. And I'm proud he's doing wonderful research, and it should reach its success because it will help a lot of people. And one of my questions is vision loss due to epithelial damage versus the photoreceptor damage. What's the ratio of these different kind of damages normally in the population? And
yeah, let me answer that first. Otherwise, yeah, you can ask your next question after I answer it. So retinal pigment epithelium damage is more in the aging population. So I don't remember exactly the number, maybe at least 10% of the all world population after they start aging process, they might get affected by various degrees from very low level to very high level. So that is the age-related macular degeneration. That is a disease caused by retinal pigment epithelium dysfunction. The other one is photoreceptor damage is mostly, it's not that common, but still there could be millions of people all over the world. But the really bad thing about the retinitis pigmentosa diseases, that is a photoreceptor damage, is usually it will happen in the disease manifestation starts very early, maybe in the age of 10 or 15. So students, and I have actually, I had a phone call from a Malayali from San Francisco. I mean, I think he came to know about me through my publication or through the website, and he asked me to talk about his daughter's situation. So she is in the University of Berkeley, which is a very prestigious university, and she is only 16 or 17, and she got this retinitis pigmentosa disease. And she's kind of like maybe 40% blind already. And he was very worried about, and he asked me about what are the therapeutics available. But after looking at her case report, I realized that she could be totally blind within the next 10 years, most probably. So that's a bad thing with the photoreceptor dysfunction diseases. It can start at an early age, but the other one is more in the aging population. So there is a big difference. Mohan, you can ask your next.
Okay, and you said the culture, in vitro culturing, of course, there will be a lot of growth factor or growth factor-like components needs to be added to differentiate into this kind of different cells, like epithelial cells or photoreceptors. Whereas if we graft them, do we need to supplement some of these things, some of these factors in order to stay there and in order to survive and in order to make a connection with the nervous neurons? Is there any components required?
Actually,
or they themselves?
Yes, that's a very question. This is something which every day I do some brainstorming on the same question you asked me. Because at this point, I mean, that's what ideally we should do. So there are a lot of limitations with the survival and integration, and we are still unable to do it because the main reason is you have to think about the area we are dealing with. The eye itself is really small size, and you are looking at a very tiny area inside the eye after transplantation, and that area is highly fragile. So, for example, retina tissue react to any kind of foreign material. Like if you get a wound or a thorn into your skin, you can see that it will develop into kind of a scar tissue. The same problem is there. Retina is highly sensitive and immunologically sensitive area, even though it has some immunological privileges. So if you transplant something, there is chance of rejection and all those things. But it's always good to, that's what we always think about. Like, for example, what you suggested, like having some factors. When we transplant it with a substrate, we can load everything into this, some of the factors into this substrate. So it can be a slow releasing. We tried the BDNF, brain-derived neurotrophic factor, with the it was coated with this transplants were coated with the BDNF microspheres, and we tried it. It helped a little bit, but the problem is, even at this point, we don't know exactly what is the determining factor. Which factor need to be supplemented to make it more live, functional, and integration. What is the real factor? Nobody knows. But if we know about it, probably there is a way we can load it into these transplants, and that might help.
Okay, thank you. One more thing. What do you expect about the cost? It could be very costly, or once it is developed.
I mean, you mean the whole research, or?
No, the patient transplantation. Once it is successful, the process of.
We haven't started. So what I'm talking about is mostly in animal models. So the stem cell products transplantation in humans the clinical trials are now limited to retinal pigment epithelium because the FDA know that by transplanting retinal pigment epithelium, there is not much adverse events happening. So it is more safe. So that's the only one now in the clinical trial, but we are now closely moving towards transplanting photoreceptors also. So these are, of course, when you run a phase 1 clinical trial, it is very expensive. So because usually the funding agency will support it for phase 1 clinical trial.
Okay, thank you.
Sir, there are some questions in the chat box also.
Let me try to find out where the chat box is.
What did I read out?
Yes, sir. The first question is there any specified dimensions of transplant in replacement approach?
Okay, can you repeat the question again?
Yes, sir. Is there any specified dimensions of transplant in replacement approach?
So you are talking, question is about humans, right? Of course, right? About transplantation.
Maybe, sir.
Yeah. So in humans, the macular area is so. That's the thing. When especially in a phase 1, the macula is the most sensitive area. So even the most expert surgeon will never try to put anything into the macula because that's the last hope for a patient. If he or she can see a little bit of light or a little bit of vision, we don't want to take the risk of putting something into the macula, and maybe whatever leftover can be even lost. So usually the transplant would be just near to the macula, but approximately, I would say that the transplants may be around less than 10% of the whole retina.
Okay, sir. The next question is there any chance of graft rejection, and do you do any typing method before going for grafting?
So in both in patients and human, in the animal models, when we use it, we use immunodeficient animal models to reduce the immunological issues. But in human patients, even with the retinal pigment epithelium transplantation, they inject the immunosuppressants into the eye, and also systemic administration of the immunosuppressants will be there for maybe at least a couple of months after transplantation to reduce the immunological issues. But immunological issues is a big problem because the graft rejection, even though not a complete graft rejection, even a little bit of rejection signs can cause scar tissue. Scar tissue, when I say scar tissue, means it's a tissue reaction, so that might also adversely affect. So these are all still challenges. So one of the techniques people are trying to do is they are trying to develop cell lines with the HLA, the immunological factor controlling component of the cells need to be suppressed. So that is called HLA deleted cells. So if that is successful, that will help to improve, resolve some of these issues.
Okay, sir. Next question. Which type of polymers are used for preparing the composites for making the retinal transplant? Is it biodegradable?
So we tried both degradable and non-degradable. So we tried the biodegradable one and non-degradable one. And with the biodegradable, there is an advantage that the substrate will not stay there. That is a good thing. But the big problem is, as I mentioned before, the retina is a very limited area. So if there is a biodegradable substrate, it needs some kind of circulation, fluid, movement, everything to clear it out. Because the retina is a very contained area, even the easily degradable substance can remain there in the environment and make it like a toxic environment, causing other adverse effects. So we try to stay away from degradable substrate. Of course, it's a good idea, but the problem is it can produce several byproducts, and breakdown products can cause some damage to other issues are there. So we are using Parylene, which is a non-degradable polymer, which so far biocompatible and as such not causing any major tissue reactions.
Next question. What are the other bioglues that can be used?
So the bioglues we use for composite grafting, that is the gluing the two components together. We tried different things, but we found that many of these things, what will happen is when you use a bioglue, it will change the weight of the cells, or the cells de-differentiate. There are a lot of reactions because of the glue. So we stay with the alginate, which shows minimum. But I totally believe that is not the ultimate bioglue for human transplantation. Probably we need to look for better bioglues, and if somebody has some suggestions, they can email me so I can try it.
Next question. Whether this grafting applicable for inborn blind people or people get vision problems during their life?
The transplantation is when you say that inborn blind people, most of the cases it is not a issue with the retina. Most of the cases it is an issue with the optic nerve that goes into the brain. So to fix that issue, I don't think we currently have any techniques, or there is not even any approaches that are feasible for congenital blindness, where because when it is a congenital blindness, even the development may not be complete. So there may not be even photoreceptors, there may not be even proper neurons. The problem with the retina is the maturation of the retina is related with the neural signals, and also there are intrinsic factors and downstream effect from the brain. Those are all involved in maturation of the photoreceptors and maturation of a functional retina. In a congenital eye, since the person is not seeing even any light, so there are no associated trophic effect and input from the brain to make the retina more functional and fully mature. So in that case, it might be totally difficult to restore vision in such people, even using a retinal prosthesis. But for the other later diseases that develop in the course of life, those are the two we discussed before. One is retinitis pigmentosa, which is mostly by mutations that affect the functionality of the photoreceptors. For that, cell replacement is an option, but so far not successful. It is still in the very early phase. The other disease is age-related macular degeneration, where the disease is caused due to RP dysfunction. We can make good retinal pigment epithelium monolayers on substrates and transplant it into the eye. But even though the approach seems to be very impressive and everything looks good, but even with that, we are not getting a beneficial effect what we are supposed to get. So I would say that it is still there a long way to go. Make it a very successful approach.
Next question. How rescue of retinal cells are done in tropic effect? Is it met in vitro and replaced after rescue?
So the question is still not clear, but I can explain about the trophic effect. So in a retina which is under the progression of degeneration, what will happen is, just like think about a plant, you are not properly nourishing and not properly watering, it will eventually start losing its leaves, and it become unhealthy, not producing any flowers, no fruits. So that's a condition where a degeneration of the retina is progressing. So it is not a sudden process. So it takes time, maybe years, for the different cells to eventually die. But before that, it will become less and less active. So again, coming back to the plant, think about you put some fertilizers and plenty of water. it might, at least some cases, even if the roots are rotten and it's unhealthy, still it might, for a couple of days, it might show some rejuvenation. The same thing with the trophy factor effect. When you inject or transplant healthy cells into the retina, because these are healthy cells, they will produce this kind of trophy factors. So one thing I forgot to mention, or I mean, I didn't want to complicate it. So there are some intrinsic factors which are also having the ability to produce trophy factors. For example, if you disturb the retina, the cells already present there, because of that mild trauma, that cells, they will produce some kind of trophy factors. So any intervention into the retina can lead to some trophy factor effect. But again, the negative thing about it is very transient. For example, a patient who shows trophy factor effect, maybe for example, the vision improvement, if you say that 1 to 10, 10 is the best vision and 1 is the poor vision. A person with the poor vision, 1, after trophy effect, he might go to 2, and that may stay for only maybe 3 months or 6 months. So it is an immediate effect that gives some happiness to the patient and some happiness to the clinician or researcher who will do the transplantation, but it's not going to stay forever.
Next question. How the integration of the transplant and the host retina occurs, and how long does it take for the integration process?
So when I say about integration, what we expect is all the neurons are connected each other in the proper way. But in a normal retina, there are at least 100 different types of neurons, and these neurons are connected to each other in a specific pattern. And that pattern is important for, so think about how complex is, an organ is capturing an image and showing it for you. So it's a highly computerized level of processing. So when we talk about integration, we cannot expect that kind of a restoration of all connections. So the neurons has a tendency to hook up between each other if they get an opportunity. So what we are seeing in our transplants are the minimum connections at the first level of connections. We don't even know how beneficial those connections are. Probably those connections are meaningless. We don't know, but we know that there are connections. That is what the question from Dr. Mohan was very significant. I mentioned that is what we are all looking for. We want to make sure that they connect properly. Only that way, there is a way the vision can be restored in a good way. Just a light perception may not be sufficient. So probably it can, I mean, it's good. It's better than nothing, but that's not what we want to see. A patient should be able to see at least some of the things, but we are nowhere close to that yet.
Next question. Taking into account the ethical issues, what is your opinion about the opportunities of research in stem cells in India and abroad?
So when we say about stem cells, I don't think there are any serious ethical issues, because there are some groups say that, oh, stem cells, it is, ethically it is wrong because it is, the cells are obtained from fetus. But it's a wrong thinking that each day fetuses are used for getting the cells. It's just like the HeLa cells for cancer cell. It was obtained from a lady long back, and it is being used all over the world by growing the cells and passing it and developing new cell lines. And there are a lot of different things that can, so same thing with that. So the initial source is maybe a fetus, but it's not like a daily harvesting of the cells from fetuses. There is nothing like that. So the ethical issue is very negligible. Then coming to the future for stem cell research in India, I don't think it is anywhere related with ethical problems, but I think it's still, I don't know. I mean, I haven't seen many papers published from India. So there are a couple of Eye Research Institute, they do a little bit of stem cells. Last year I was in PGI Chandigarh. There is a group there who work with the stem cells. There are a couple of other centers in South India also, but still it is it's a very very early stage. But the reason I don't know exactly why, but I think, as I mentioned before, stem cell has great potential not just for eye diseases, but I think it has a lot of therapeutic applications. Even many of you might know that even for cancer, so it's a very fast-growing field for cancer treatment. So because you can manipulate stem cells and use it for different purposes, that's a good thing.
Next question. Can diseases like color blindness caused by reasons like vitamin A deficiency be treated with this technique?
No. So anything with so that is you have to supplement with the vitamins that causes color blindness. I don't think stem cell is a suitable approach for that.
Thank you, sir. Any more questions? Okay. Thank you, sir, for answering the questions of the participants. I request all the participants to find the feedback form provided in the chat box and to fill the form as well. Now, I welcome Ms. Vishnupriya Mohanan, student representative, to deliver the vote of thanks.
Am I audible?
Yes, you are.
Good morning, one and all. On behalf of Postgraduate and Research Department of Microbiology, Sri Shankara College, I take this opportunity to extend my sincere gratitude to our resource person, Dr. Biju B. Thomas, for sharing your excellent lecture. The session was informative and enlightening. Thank you, sir, for spending some of your valuable time with us. I also thank all the participants for your cooperation and active participation throughout the session. Once again, thank you all.
Thank you, Vishnupriya.
Thank you
Now
very
that